Hello, everyone. Hope by the time of the video you guys are watching, you guys are, physically and mentally doing well. I know what's happening right now in Lebanon is quite hard. It's actually quite heavy on all of us, and it actually makes kind of these small tasks like studying or working or any of these kind of things quite mentally challenging. Like chaos is happening outside of us, and we need to basically maintain calm and actually focus. So I know that this is not easy, and I really applaud you for really keeping this up. So saying that, I also want you guys to take full advantage of this, open hours, this kind of open office hours. This means that if there's something that you guys do not understand in my recordings or my lectures or whatnot, please send me an email and schedule a Zoom meeting. I am free, as I said multiple times, whenever you guys want me to be free. You guys are paying so much money for LAU, and basically, of course, some money is being allocated to my salary for you guys for to do my job, and that's one of my actually jobs. Is to make sure that you guys are following up, and I'm helping you as much as I can. So please use that to your advantage. All right. Now, putting that aside, and having said that, let's go into chapter five. So, in chapter five, we will be talking about membranes, and in specific, the cell membrane. The cell membrane is, the first structure we see or realize actually in a cell, and it has a very, very crucial, job. It helps to separate the outside from the inside of the cell. So it creates this nice physical barrier that, kind of helps the interior of the cell not be exposed to harmful substances that may be found on the outside. So that's one of the biggest functions the cell membrane has, but it has many, many different functions. And one thing, of course, that we were explaining a lot is that tell me the structure and I will tell you the function. So to understand the functions of the cell membrane fully, you really need to go into the membrane structure. So that's where we're going to start off today with the structure. So this figure should be quite, familiar with you guys, specifically to chapter three, where we were talking a lot about macromolecules and specifically lipids. So a form of lipid that, we see a lot in the membrane is phospholipids. So these phospholipids are these glycerol, which are these three carbon backbones that have alcohols, so they have polyalcohols in them. And through dehydration, you get these two fatty acids connecting to two of the carbons through the oxygen. And then you have in the third carbon, you actually have a phosphate group with a choline. So if you look right now just at the fatty acids, you can see they're all covered in carbon and hydrogen. And one thing that we know with carbon and hydrogen is that they are electronegatively very similar to each other. So once they create a bond, they share that electron quite well, quite fairly. So you're not going to get these partial negative or positive charges. So you're going to get non-polar, non-polarity, so hydrophobic, they're going to not like water. So they don't want to do this hydrogen bonding with water at all. So they're called, these fatty acids are very, very hydrophobic, so non-polar. But if you look at the phosphate, it has a lot of oxygens. And oxygen is a very strong electronegative element, meaning that it actually has a very strong affinity towards an electron, meaning that when it actually creates a covalent bond with, another element, it's going to really kind of pull the electron more to its side, and it's going to create this unbalance. So these partial negative and positive charges. Hence, it's going to give, polarity, and it's going to be very hydrophilic, meaning it's going to love water. So you see that already from the phospholipid, you have kind of these kind of properties of both hydrophobic and hydrophilic. Hydrophobic coming from the fatty acid tails, and hydrophilic coming from the phosphate head. And what this gives you is that when you throw phospholipids in water, what they're going to do is they're going to assemble in a bilipid membrane or bilipid layer, meaning that you're going to get two layers of phospholipids because the phosphate will create a barrier from the upper side and the lower side, and this will prevent any water to get inside to the hydrophobic tails. That's why you have basically a bilipid layer to create this barrier that not allowing any water to go into, because the lipids hate water and also water hates them. So that's why you get this bilipid layer. So this lipid bilayer, sorry. So in this lipid bilayer, you actually have proteins that are actually inserted actually into this phospholipid bilayer. And at the beginning, people were thinking that, okay, they're inserted there and most probably they're just stuck there. They don't move. But that's not true. It was actually 1972. It was Sir Jonathan Singer and Gareth Nicholson that proposed the fluid mosaic model. And this fluid mosaic model, what it basically entails is that the lipid layer or the phospholipids and the proteins are actually quite, they're moving. They're quite fluidy. They're not static, they move around. So they propose that the mosaic of proteins floats in or in the fluid lipid bilayer, like boats on a pond. So what they're saying is that the proteins are like boats in a pond, which are your lipid bilayer. That they're not static, they're actually just floating around the lipid layer. So the phospholipids by themselves move, and the proteins that are inserted there also move. That's what this basically this fluid mosaic model actually means. So if we take a look actually right now at the cellular membrane, at the main components of it, there's four. One of them is the phospholipid bilayer, which is consisting of the phospholipids, but it also consists of something called sphingomyelin. So I'm gonna basically, bring here your textbook. So give me 1 second. Here you go. So this is your phospholipid, as we were showing you. This is here your sphingomyelin. Very similar actually to your phospholipid, right? So basically it has the two lipid tails. It also has the phosphate head. But look at this guy over here. It doesn't have this three carbon, so it doesn't have the glycerol. That's changed. So that's why they're different. They're very similar in structure, as you can see, these two nice little tails with the phosphate head, but they're quite different in, basically in the molecular formula. So keep that in mind. So the phospholipid bilayer, as I was saying, it consists of actually, the phospholipid, the sphingomyelin, and also cholesterol. Cholesterol is within this phospholipid bilayer, and we'll be talking about that a bit later. Then you have proteins. You have proteins that are actually, spamming or actually going through the membrane, and those are called transmembrane proteins. So membranes that are going through the membranes, or membranes that are basically attached to the phospholipid bilayer, meaning that they're not spanning it, they're just attached to it. And these are called interior protein networks. And we'll talk about them more in details. And you also have something called cell surface markers. These are glycoproteins and glycolipids. In chapter 4, we were talking a lot that the Golgi, when it accepts lipids or proteins from the ER, it's gonna sometimes, of course, fold them correctly, but it's sometimes gonna put some carbohydrates on them. And these carbohydrates actually, serve as an identification. It's kind of مثل هوية, like an ID, actually, because it actually gives uniqueness or an ID to the cell. And I'll basically use this as an example. So you definitely guys have heard that people have, group A blood type, group B blood type, or AB or O. Well, this is actually glycolipids. So group A have a certain glycolipid, group B have a certain glycolipid. So they're giving there an identity. So that's what surface markers are. So if we take a look right now at this whole cell membrane, this is how it looks like. So you have the phospholipids, basically here you also have sphingomyelin, and you have cholesterol, which is shown here in green. Then you have basically these transmembrane proteins that we also like to call them as integral proteins. So integral proteins, just like transmembrane proteins, so the same thing, they are proteins that protrude through the plasma membrane, meaning that they go through the plasma membrane. The peripheral proteins, as I said, these guys are so the ones, the interior protein networks, these are the ones that are attached either to the outer or to the inner membrane, but they're not passing through it. That's the difference between peripheral and integral. So keep that in mind, please. So that's basically how the whole actually thing looks like. Of course, you have the glycoproteins and the glycolipids. These are basically kind of these identifiers of the cell. And of course, things around here are moving, hence the fluid mosaic model. They're not static, things are moving around. So, how do we study the cell membrane? Well, there are two main ways of doing so. The first one actually, consists of using TEM, this transmission electron microscope. And what you do is you get the cell membrane and you actually embed the cell membrane in basically this epoxy matrix that is at the beginning quite, it's like very kind of gel-like, very, kind of a waxy substance. You put your cell membrane and then you let it dry and it becomes this hard, basically, substance. Once it becomes hard, you start cutting it into 1 micrometer thick. Because if you remember, transmission electron microscope requires that your specimen be very, very thin, and to the point of 1 micrometer thin, even almost. So you basically cut that, and then you basically pass electrons through it. And of course, depending on the density, you start to get actually an image. So here is an example of basically how it looks like. This coloring is called deep, this is called fake coloring, meaning that this is not true. It's the computer that's giving this these certain colors. So based on the density, it gives it a color. Like you have the, for example, here, this is the phospholipids. You have the first layer here and this here. They're the same, and hence it's giving actually, orange. While the lipid tails or fatty acid tails, are giving a certain density and it's coloring it differently, and hence also, the outsides. So here you actually get a really nice, beautiful images, which allows us to actually really study the structure of the cell membrane. So that's one way. Second way is basically a bit different. Here you take the whole cell, you put it in a medium. So this medium basically contains, certain substance or liquid, and then you freeze it using liquid nitrogen. Once you freeze it, you take a knife and you basically kind of crack it. You kind of poke and you crack it. And because it's frozen, what happens with the phospholipid lipid bilayer, it cracks in half. So basically, now basically you have this exposed lower half of the lipid bilayer and the other basically fractured upper half. So you're basically breaking this bilipid layer into two. So then you take basically the one half and then you can do SEM. So this scanning electron microscope, and you get this really beautiful picture. So this is basically how we can do either through the TEM or this freeze fracture microscopy. So now let's go again into the phospholipids. So again, this is repeating these things. Phospholipids are consisting of glycerol, a 3-carbon polyalcohol, and 2 fatty acids that are attached to the glycerol, and a phosphate head. The fatty acids attached are the nonpolar hydrophobic ones because they have a lot of carbon and hydrogen. The phosphate, which has a lot of oxygen, is the one that's polar and hydrophobic. Hence, phospholipids are both hydrophobic and hydrophilic, meaning when you throw them in water, they will create this, spontaneously create this bilayer because you want to protect the fatty acid tails from water. So you create basically a upper and a lower layer to protect any water to enter the fatty acid tails. So literally, you fill the phospholipids and spontaneously you create this bilayer because of these properties. Please keep that in mind. And as we said, the bilayers are quite fluidy. Things are moving around, but they're not. They are fluidy, but not very fluidy. Because imagine if they're very fluidy, they're super fluidy. That if I just literally kind of blow on the cell or just touch it a bit, it's gonna fall apart because it's just fluidy, right? That is not the case. Because of the phosphate heads are quite polar, they're going to create a lot of hydrogen bonding with the water surrounding it. And this basically creates this adhesion. So you create a lot of surface tension. And this surface tension keeps the cell quite intact, actually makes it quite strong, but not strong enough to keep it super static. Because again, sure, these a lot of in general, the hydrogen bonds are weak. A lot of them make it stronger, but they're not super strong, you know. So they're strong enough to basically keep it intact, but not strong enough to keep that everything static. Things move. That's what I mean. You know that they're strong enough to keep the cell, in one piece, but not strong enough not to make anything move. So everything's things are still moving because it's not as strong as you'd kind of expect. So of course, at the beginning, you start let's apply this fluidity thing in scientific thinking. So let's imagine you're back in time and you're a scientist and you're thinking, okay, is this phospholipid bilayer, is it actually rigid or is it actually fluid? So you have a hypothesis. You say, you know what? No, the plasma membrane is fluid. It's not rigid, meaning that it basically it moves. So the prediction is, if the membrane is fluid, hence the membrane proteins can diffuse laterally, meaning they can move around. Let's test this. Let's get a human cell and actually label all the proteins in blue. Let's get a mouse cell and label all its proteins in red. Let's fuse these cells together. And if my hypothesis is correct, you should basically have the mixing of these membrane proteins all over the cell. If it's rigid, the blue proteins will stay on the right and basically the red will stay on the left. That's what it's supposed to be. However, when you put them, you see that over time, the basically hybrid cell shows increasing intermixed proteins because the proteins are able to move, they're mixing. So your conclusion is at least some membrane proteins can diffuse laterally in the membrane. So this is one experiment. But if you guys remember in what I was really saying in chapter one, the more experiments you have, the more valid your hypothesis is, right? So this one experiment might, show that our hypothesis is correct, but it doesn't make it, quite 100%. Because what if newly synthesized proteins were inserted into the membrane during the experiment? Meaning, what happened, of course, when you create this big cell of the fusing, so when you get the human and mouse cell fused together, this cell still can produce new proteins, and they can be inserted randomly, so giving you this intermixed membrane. So this is not really moving of proteins, it's just newly formed proteins being inserted randomly around. So this is something you need to control. So me as a scientist, what I would do as my next experiment to fully prove this is that I would take this cell and I would fix it, meaning that I would actually stop its protein translation. And you can do that because then no new proteins are synthesized and you really can just attribute these mixing to movement and not to new proteins being formed. So keep that in mind. So one thing about the bilayer that we really need to start looking into is its fluidity and what influences it. So the majority of these kind of fatty acids that are found in the phospholipids, they're saturated, meaning that the carbons are actually do not have double bonds. And because they do not have double bonds, this makes them less fluidy. Because if they were unsaturated, meaning that they had double bonds, they would be very, what's it called, liquidy and very fluidy. So that's one thing that actually prevents the whole cell membrane to fully become very fluid. And because you don't want it, you want it to be fluid, but not too much, because that could really create some issues, and I'll be talking about that later. But because of this, warm temperatures can also influence fluidity. Other than being, of course, the saturation and unsaturation, temperatures can affect it. Because when you apply warm temperatures, you're giving energy. Because again, heat is a form of energy, and this is gonna make the the lipid tails move around much more. If and vice versa, if you basically apply cold temperatures, so you remove heat and energy, you will actually make them quite rigid. So they become less fluidy. So of course, there we need to find a way to actually counteract this. And bacteria have found the perfect way of doing so. Bacteria have these enzymes that we call fatty acid desaturases. So the name is desaturases. So they desaturate, meaning that they will actually add, these double bonds to the carbon. And this creates from it goes from saturated fatty acids to unsaturated fatty acids, and unsaturated fatty acids are quite fluidy, and this can counteract the cold because as we said the cold makes things rigid. Well, let's counteract this by making the fatty acids more fluidy by saturating them using the fatty acid desaturators. That's one way of doing so. So if we look at the lipid composition, you really can say a lot about actually, the structure. An example is here between the ER, the Golgi, and the plasma membrane. So as we know, the ER and the Golgi, they are also membrane bound organelles, meaning that they have a membrane. But the membrane is actually quite different to the plasma membrane. And this is interesting, even though because most of the phospholipids or lipids are actually formed in the ER. So you kind of think that they might be the same, but actually they're not, weirdly enough, in my opinion. But anyway, if we look at actually the plasma membrane and we compare it to the ER membrane, the plasma membrane is actually mostly composed of saturated fatty acids. The ER is not, it's mostly composed of unsaturated fatty acids. And the plasma membrane has much more cholesterol compared to the ER actually. And this gives the plasma membrane more bulkiness actually and makes it more thick and actually less fluidy compared to the ER membrane. And this explains a lot why the membranes of the ER is much more folded because it's much more fluidy. It's able to fold. The plasma membrane is not able to fold that much because it's not that fluidy. It is fluidy, but not as much as the ER. Because the ER it has more unsaturated fats and less cholesterol, so it is less bulky. So it's easier for it to actually fold. So, knowing the lipid composition can really help you actually understand the folding, the thickness, and the shape of the membrane. So now that's done, let's now talk more about the membrane proteins. So these are the proteins that are found in the membrane. And there can be many different types. There can be, for example, proteins that are helping transporting things from the outside to the inside. These could be membrane proteins that are actually creating or helping kind of chemical pathways like enzymes. They actually catalyze chemical reactions. These could be also cell surface receptors. So these are proteins that actually are very sensitive to the outside and they bind to certain, signals that actually alert the cell. Or they can be the cell surface identity markers. And these are kind of form this kind of, let's say, ID and kind of help the cell be identified. Or they can be cell-to-cell adhesions. So these are the ones we actually spoke about in chapter 4. So these are proteins that are bound in their membranes, and they basically once they basically bind with each other to kind of help the cells to adhere together or connect with each other. Or they can be basically these kind of membrane proteins that attaches to the cytoskeleton of the cell. So that's one way of also. So as you can see here, you have a lot of different functions. But because you have a lot of functions, you have a lot of different structures, correct? Because again, structure gives function. So you have many different functions, hence you have many different structures. But one thing that we also find in these membrane proteins that even though they have many different structures and hence many different functions, one function they all share is that they are in the membrane, and hence they have similar structures because of that. And we will talk about two different membrane proteins, the ones we were talking about. These are peripheral proteins and basically the integral membrane proteins. So peripheral proteins are the proteins we spoke about before. These proteins do not span the membrane. These proteins are just attached to the membrane. So again, I'm going to go back to make this more understandable for you guys. So these are the peripheral proteins. They're attached to the membrane. They are not spamming or going through the membrane. Those are integral proteins. So keep that in mind, okay? So how do integral proteins actually, attach to the cell membrane? Well, two ways. Either peripheral proteins actually, will attach to the integral proteins, and that's how basically they interact. So sorry, peripheral proteins that can interact or attach to the cell membrane through two ways, either through interacting with integral membrane proteins, or they're covalently attached to special lipids that we call anchoring molecules. So these are basically anchoring molecules. These are special lipids that have a hydrophobic tails that are able to actually, insert themselves into the cell membrane, and they are able actually to kind of chemically bond to the proteins. So to these basically peripheral proteins. So think about this as as kind of like an anchor. So hence the name. Basically, you have the boat, which is the protein, and basically you have this anchoring molecule that are anchoring it here to the dock, to basically the parking station. So again, repeat this. Peripheral proteins can attach to the cell membrane to two ways. The first way is either to attaching to the integral membrane proteins or covalently attaching to the anchoring molecules. So integral membrane proteins are a bit different there because these guys have to go through the cell membrane. The only way you're able to penetrate the cell membrane is if you have basically hydrophobic areas. Not basically to enter an hydrophobic area, you need to be hydrophobic. So that's one thing. So that's what integral membranes proteins have. They have these transmembrane domains that are actually made up of hydrophobic amino acids. So basically, these hydrophobic amino acids are the ones that are spamming the lipid bilayer or basically entering it. And let me show you an example. So here you have basically this transmembrane proteins or these integral proteins. You have basically basically here in red. These are the polar amino acids because they're attaching to the water and basically here. But then you have here the blue part, and this is the transmembrane domain. And this is all hydrophobic amino acids because they're the ones that can actually interact here with hydrophobic fatty acids. And in this case, they're actually, found in this nice alpha helix structure. And this is what helps the protein actually, go through or spam the membrane. One a protein to actually go through the membrane needs just one transmembrane membrane domain, but sometimes you can actually have seven. You have one, two, three, four, five, six, or even seven. And how many transmembrane domains you can actually divide this into special receptors. This is something you guys will definitely learn much more in pharmacology, but not now. However, sometimes these transmembrane domains, other than alpha helices, they can be actually, beta sheets. And this creates basically these kind of beta barrels or these pores. And this kind of a pipe, it really kind of resembles a pipe, it kind of, creates this kind of pipe kind of structure in the middle of the membrane. And what it does, it allows water and small polar molecules that could not go through the hydrophobic areas actually now pass through. So I'm going to stop here. This is the first part. The next parts I'll basically give them later. The whole reason behind I'm stopping is that I want you guys to take a break. I found out that putting too much information in one video doesn't help anyone. So I'm just going to make them quite small and short.